Good afternoon, everyone. I'm Robin Gasser, the Associate Dean Research of the faculty. Welcome to the Dean's Research Seminar. Before we begin, we acknowledge the traditional owners of the land on which we gather, and we pay our deep respects to elders past and present. Today we have the Dean, Professor John Fazakerley, presenting together with Julio Rodriguez-Andres, and they're giving a lecture on mosquito-borne virus infections. John is a virologist with a keen interest in One Health and virus diseases of major medical and socio-economic impact. He's known internationally for his work on viruses transmitted by mosquitoes, flies, and ticks. In his early career, he was at the University of Durham, and also at the St. Thomas Medical School, as well as King's College in London, University of Pennsylvania, Scripps Research Institute, and the University of Cambridge in the UK. After that, he spent 15 years in the College of Veterinary and Medical Sciences at the University of Edinburgh, where he was chair of virology and group leader at the Rosalind Institute. And from 2011 to 2016, he was the director and CEO of the Pirbright Institute, a world-leading centre for research, surveillance and control of viral diseases of livestock and viruses that are spread from animals to humans. He's also held a number of grants from the UK bodies, including MRC, which is the Medical Research Council, BRC, Biotechnology and Biological Sciences Research Council, and also the Wellcome Trust. And he's been the coordinator of two large international EU programs on virus, vectors, and chikungunya virus. Professor Fazakerley is a fellow of the Royal College of Pathologists and the Royal Society of Biology in the UK. And he's been the editor of Journal of General Virology, executive editor of Neuropathology and Applied Neurobiology, and he's chaired a number of large committees working in different areas on major aspects dealing with vaccine production and viral diseases. John joined the University of Melbourne in 2016 as Dean of Veterinary and Agricultural Sciences, and he's also Professor of Virology at the Peter Doherty Institute for Infection and Immunity. Today, we're fortunate to have Dr. Julio Rodriguez-Andres to join him in giving this presentation today. So welcome to both of you.
Thank you, Robin. Thanks for the kind introduction. Okay, I will try and do my screen sharing and we'll get going. Okay, hopefully that works and you're seeing the presenter view on that. So I'll introduce Julio a little bit later, but as Robin just said, we are based in the Peter Doherty Research Institute, and I have an adjunct position in the Faculty of Medicine, Dentistry and Health Sciences. So here's our research group in the foyer of the Peter Doherty Research Institute, and this is what I'm going to talk about today. I'm going to talk about arboviruses. What are they? Why are they important? And what are the research questions that those of us that work on these viruses are interested in? I'm going to outline some current research projects on arbovirus encephalitis and show you a bit of data from our current projects. I'll wave my hands at some of the results, but the idea is that you get an understanding of the kinds of experiments that we do. Julio will then join me to summarize our recent work on mosquito immunity, which provides a major breakthrough in understanding in that particular area. So arboviruses, what are arboviruses? Well, they're arthropod-borne viruses. So a lot of you might ask, well, what are arthropods? Well, arthropods include mosquitoes, flies, midges, ticks, and sea lice. For example, ticks are not flies, and therefore we have to go to the next phylum of taxonomy up, and that is the arthropods. So the word is arthropods, and arthropod-borne viruses is abbreviated to arboviruses. So these kind of viruses are important in both medicine and veterinary medicine. And here's a list of some of them that you might recognize some of the names, and I will talk about some of them. The ones in black are transmitted by mosquitoes. The ones in blue, dark blue there, are transmitted by ticks. I've put the light blue one in, southern elephant seal virus, just to make the point that there are arthropods of the seas, in this case sea lice, and this elephant seal virus is transmitted by sea lice. So similar things are probably going on in the oceans that are going on land. And blue tongue virus is a virus that's transmitted by midges. There are also viruses that are transmitted by flies, other than midges and other than mosquitoes. So how does this transmission work? Well, let's start at 12 o'clock. And so you've got your arthropod, which is a mosquito or a fly or a tick, and they've got a virus infection. And they find a vertebrate, it might be a bird, it might be a mammal. They bite the vertebrate and they inject a little bit of saliva at the time that they bite, and in with the saliva goes some virus. And then they suck out a whole load of blood, and that's what the mosquito or the tick or the fly wants, a blood meal. And then the animal has a high replication of virus within its tissues, which results in a lot of virus in the blood. So very high levels of virus in the blood is what these viruses have become selected to achieve. And they need to achieve that in order that when another arthropod bites the bird or the mammal, that arthropod will take a blood meal that contains sufficient virus to transmit the virus back to the arthropod. The arthropods, we're now on the bottom at 6 o'clock here, the arthropods will then bite another vertebrate and transmit the virus to another vertebrate. Those vertebrates will be bitten by another arthropod, and so the whole thing goes round. And this is how for probably millions of years these viruses have stayed in existence. They are going from the arthropods to mammals and birds. Now, there might be unusual vertebrates in some cases. Here's an alligator. Alligators have thick skins, yes, but they still are susceptible to mosquito bites around the eyes and around the mouth, for example, and alligators are found to be infected with, for example, West Nile virus. So they might be important in the transmission of these kinds of infections. But quite often, animals such as that are dead-end hosts. They don't necessarily take part in the transmission cycle because they don't produce enough virus in their blood. And that's often the case with humans. For example, humans sitting on this lawn here might be bitten by a mosquito. Although some people might get sick, the majority of people won't have sufficient virus in their blood to really be part of the transmission cycle. And the same is true for horses. And from the point of view of transmission, we call these dead-end hosts because once the virus gets into these, it's not really going to go anywhere else. But sometimes humans can be part of the transmission cycle. They can have a lot of virus in their blood. And then when an arthropod bites them, that arthropod gets enough blood to pick up some virus and then transmits it back to another animal. And sometimes that can be another human. And in fact, some viruses go around human to human transmission. The best known example of this would be dengue. And I'll talk a little bit about dengue in a moment. Here's one that you may remember. So this is Zika virus. This was a problem in 2015, 2016. It's not such a big problem at the moment. There are still a few little outbreaks of Zika virus simmering in different parts of the world. But in 2015, 16, this was in the headlines a lot. And you may remember that people were very worried about pregnant women giving birth to babies with hydrocephalus. And this is a feature of many of these arboviruses. They go for the brain and they cause brain pathology. So here's one that's in the news at the moment, Japanese encephalitis. And this is a virus that is found in water birds, actually particularly herons and egrets. And from the water birds, mosquitoes transmit it to pigs, horses, and humans. And these are particular kinds of mosquitoes, in this case, Culex mosquitoes, in this case, Triteniorhynchus Culex mosquitoes. The pigs and horses usually get subclinical infections, although pigs can get abortions. And the majority of human infections are also subclinical. And Japanese encephalitis is the leading cause of human viral encephalitis in Asia, with 30 to 50,000 cases reported annually. And this is probably an underestimate. Unfortunately, when people get seriously ill, they get acute encephalitis, and this results in paralysis, seizures, coma, and death. And of young children getting Japanese encephalitis virus that would result in acute encephalitis, about a third of these survive. A third of them survive, but with neurological sequelae, and a third of them die. There are vaccines for Japanese encephalitis virus. There's an inactivated vaccine, a rather controversial live attenuated vaccine, which comes out of China, and a more recent vaccine based on genetic modification of the yellow fever 17D vaccine. So Japanese encephalitis has been in the news recently in Australia here because there have been cases of it. And this is taken from the last couple of weeks. And you probably heard about this on the news, or you will have seen it in the newspapers or on the television or on the websites. So there's a lot of publicity out there at the moment. What is Japanese encephalitis and why is it spreading in Australia? So in the last month or so, Japanese encephalitis virus has been discovered in more than 40 piggeries across Victoria, New South Wales, Queensland, and South Australia. And so far, we've had 2 human deaths. And human cases have not previously been detected this far south in Australia. And here's a baby who was in the news headlines over the last couple of days, who's in hospital, seriously ill, having Japanese encephalitis virus. And it's often the babies and children that get the most seriously ill. This is a publicity poster from the state of Victoria. Beat the bite. Do you know mosquito-borne diseases can make people ill? Here's one from a little further back, June 2000, when the big issue was a virus that had suddenly appeared in New York. Hence the front page of this magazine from a British newspaper. And as you can see there, it says, out of Africa, the deadly terror stalking New York. And this was first brought to prominence when birds started dying and some animals got sick. And the vets in New York Zoo realized that the birds and some animals were getting sick. And they actually managed to isolate from this a virus called West Nile virus. And as the name of this virus suggests, it actually comes from the Nile River Delta in Africa, or was first isolated there, but it is found in a number of different areas, but it hadn't previously been found in North America. To cut the story short, it turns out it probably came from Israel on somebody who came in on a plane who was viremic, who was probably then bitten by a mosquito. The mosquito bit some birds. The birds transmit amplify West Nile virus, and West Nile virus usually goes between mosquitoes and birds. But it also ends up in horses and humans. And actually, there were cases of horses quite quickly on the eastern seaboard, and then human cases. And the virus spread over the next couple of years right the way across the United States and is now endemic in the US. So here it is, West Nile fever. It's transmitted by the Culex species mosquitoes, the same ones that transmit Japanese encephalitis. It's found around the world, and here in Australia, it's called Kunjin. And its natural cycle, as I said, is mosquitoes and birds. In humans, generally, most people get no symptoms, or maybe 20% of people are mild febrile illness, and then a very small percentage, a bit like Japanese encephalitis, get severe disease. And again, this can include encephalitis. This is another one, equine encephalitis, which is found in the Americas, in eastern, western North America, and in Venezuela, in South America, and countries near there. It's found in tropical forests and swamps in the Americas, and it's transmitted by mosquitoes that like to live in these forests and swamps. And actually, rainy seasons can result in changes in mosquito populations, and big outbreaks of this tend to require the right kind of mosquito and viruses that have got a few changes in their genome. But when the outbreaks get going, they can result in hundreds of thousands of equine cases and tens of thousands of human cases. And the public health people know that a big epidemic is coming when the horses start to get sick and then the humans start to get sick. An example would be in 1995 in Venezuela and Colombia, where there were 85,000 human cases, and 3,000 of these had neurological symptoms. That is, again, they had encephalitis. This is spread not only by Culex mosquitoes, but also by Aedes mosquitoes. And you can tell the difference here. The Aedes ones are the stripy ones. Which brings me to the Asian tiger mosquito, so-called because of its stripes. This is a map showing the distribution of the Asian tiger mosquito. Well, this mosquito started here in Southeast Asia, which is why it's called the Asian tiger mosquito. This is its natural habitat. And it's been spread around the world by global trade. The larvae of the mosquitoes thrive in the water, which is inside tires, which are distributed around the world, used tires. It's also in things like lucky bamboo plants that get shipped around the world. The larvae are in little pools of water, and then they hatch when the lucky bamboo plants reach some other part of the world. Anyway, this virus has now spread around the tropics throughout the world, and you can see its distribution here in red. And why this virus is particularly important is it likes biting humans. So it's particularly important for human health. It likes biting humans either at night or indeed in the daytime as well. And with climate change, the Asian tiger mosquito, Aedes albopictus, is likely to actually move further out of the tropics. So if we look at Europe here, the prediction is that it will move much further north in Europe, even reaching the southern parts of the United Kingdom here. So climate change is making a difference, or is predicted to be making a difference. And here are two of the diseases transmitted by this particular mosquito, dengue, which is probably the most important of the arboviruses, and chikungunya. Actually, dengue and chikungunya are a differential diagnosis from each other, and it can be quite difficult to tell them apart. Dengue is found in the tropics and the subtropics worldwide. It's endemic in over 100 countries, and half the world's population is estimated to be at risk, with infections of around 400 million a year, and 25 of these have some kind of symptom. Chikungunya, on the other hand, has been considered to be more epidemic, although it's not entirely clear whether cases previously called dengue might have been chikungunya. But chikungunya came to prominence in recent decades in 2006, when the French island of La Réunion had over 200,000 cases. And this happened all within a couple of weeks. And the health service on La Réunion was overwhelmed, and a lot of people became seriously ill, and 254 people died. And since 2005, chikungunya has spread into India, Indonesia, the Maldives, Myanmar, Thailand, where there's been over 2 million cases. And then it hit the Americas in 2013, where there's also been over 2 million cases since 2013, mostly in the Caribbean. I worked on this virus for a good while and led a big international consortium, probably the world's largest consortium looking at chikungunya virus, and we developed diagnostics for it. We understood the pathogenesis of it, and we developed a vaccine. But I'm not going to talk about chikungunya today. So arboviruses, what are the general questions that those of us working on these viruses have? Well, they are, how are these viruses transmitted by mosquitoes? Might sound straightforward, but when you listen to Julio shortly, you'll see that it's not totally straightforward. How do they cause diseases in vertebrates? What kind of diseases do they cause? Well, generally joint disease, brain disease, or haemorrhagic fevers. How should we treat these diseases? Do we have antivirals? Not many. How can we prevent them? Mosquito control has been a key thing, but also vaccines for some of them. And how do we detect them? And how do we detect new and emerging viruses, such as Zika, or viruses that emerge in new regions, such as Japanese encephalitis, which is now in the southern parts of Australia. I'm not going to address all of these issues today, but I wanted to bring up some questions here so you can see the kinds of things that people working in this field are interested in. And this is truly a One Health issue, because as I've already shown you, a lot of these viruses go from animals where they may or may not produce disease to humans where they may or may not produce disease. And understanding that is important to both veterinary medicine and human medicine. So I want to talk about two things now before I hand over to Julio. So this is now the second part, and I said I'd give you a brief overview of some of the things that we're interested in the laboratory. And we're going to talk about two things. I'm going to talk about disease in vertebrates, and Julio is going to talk about virus transmission by mosquitoes. So if we're going to study this experimentally, it's best to study something that's not going to make humans sick. And Semliki forest virus has long been such a virus. This is a virus that was isolated from Uganda, the forest in Uganda, and the forest is actually called Semliki, and the Zika forest and the Bunya forest. There's a famous virus called Bunya virus, famous group of viruses actually, and there's a virus called Bunyamwera virus. But anyway, the Semliki forest virus, the Zika forest, and the Bunya forest are all in Uganda, and these have each had viruses isolated from them and named after them. The Semliki forest virus is closely related to chikungunya, which I mentioned, to Ross River, which is an Australian virus, which causes painful joints and is transmitted by mosquitoes here in Australia, and has its reservoir in macropods, that is the kangaroos and associated animals. And there's also a virus called Barmah forest virus, and the Barmah forest is on the Murray, and this also causes joint disease. The Semliki forest virus is well used in laboratories because its molecular and cellular virology are well understood. It also can be used in level 2 laboratories, which means that it doesn't cause serious human disease. But in mice, which is the main laboratory animal for studying such diseases, it causes encephalitis. In fact, a subacute demyelinating meningoencephalomyelitis for those that are into pathology. So it's much studied as an experimental model of virus encephalitis, both acute encephalitis and fatal encephalitis. Not all strains of the virus are fatal in mice, some are, some aren't, which is an interesting thing in its own right. So this is our experimental workhorse, or one of them, Semliki forest virus, and the one I'll talk about today. It's an alpha virus, as is Ross River and chikungunya. And these alpha viruses can be easily genetically modified, which is a major advantage to us. And over the years, we've made many genetically modified viruses. Let me talk a little bit, though, about virus genetics, because it is something that you hear about in the news these days with messenger RNA vaccines, for example. So here's a messenger RNA molecule. It's a molecule made up of 4 different bases, and it is designated like this as a 5' end and a 3' end with a little circle on the 5' end, so we know which is the orientation of the molecule. And these molecules code for proteins, which are the functional workhorses of cells. They provide the structure of cells, and they provide the enzymes that generate energy, for example, and allow the cell to function. So all of our proteins are encoded in the genome, and the thing that is between the genome and the proteins are these RNA molecules. And at the end of the RNA molecule is a polyadenylation sequence, a poly-A sequence. And this is what we call a messenger RNA. So you'll have heard about mRNA vaccines for coronavirus. And in the case of a coronavirus, the vaccine might have a structure similar to this, and the box would contain the sequence for the spike protein of the coronavirus or some other protein. Alpha viruses are very simple. They basically have that kind of structure. And here are some chikungunya viruses that we've made over the years, and some other ones. And you can see that in this case, we've added in a molecular marker, something called ZS green, so that when this virus gets into a cell, it will make this marker and turn the cells that it infects green. That's quite useful. We can also do things like duplicate the subgenomic promoter of the virus. I won't get into why we've done that. We can also put in biochemical markers. These produce, and this is an enzyme called luciferase. And when the virus gets into the cell, the luciferase enzyme is made, and we can measure the amount of that enzyme, which then becomes a proxy for how much virus replication is occurring in a cell and gives us a quick assay for that. So another good thing of these viruses is they're very easily manipulated. And this is what happens when we put a green virus into a mouse. So here's our mouse. We inoculate it with virus, and then this is the brain taken out of the mouse, and we're looking down on the surface of the brain, and you can see that the virus has got into these different regions, and we can tell where it is because it's fluorescent green. And if we have a section of the brain, we can see these different cells that are infected with the virus. So let's see what happens when we take our laboratory mouse. And I should say, for those that don't think that we should be doing experiments on mice or other animals, it's pretty difficult to study virus encephalitis in anything other than a experimental animal, since nobody wants a brain biopsy. You can actually look at virus infections in many other tissues, in humans or horses, for example, by taking, let's say, a skin biopsy if you were interested in the skin, or perhaps even a liver biopsy if you were interested in the liver. But nobody nobody wants to have a biopsy of their brain, and nobody wants to biopsy the brain of, for example, a horse. So all of the work that's been done on encephalitis, or much of it, has been done in experimental animals, and the experimental animal has generally been the mouse. So here's our mouse, and normally the infection of an arbovirus would be via a mosquito bite. We use a syringe and we inoculate the virus into the peritoneum of the mouse, the abdomen. And what we have here is a titer of virus on the left-hand side on the y-axis. And this is in logs. So 3 means 1,000, 4 means 10,000, 5 means 100,000, 6 means 1 million, 7 means 10 million, and 8 means 100 million. 100 million what? 100 million infectious units of virus. So when the virus is given into the mouse, you can see that very quickly, within 24 hours, there are 100 million infectious units of virus per ml of blood. That's the huge amount of virus that the virus needs to achieve, so that when the vertebrate is bitten by another mosquito, the virus will be transmitted back into that mosquito, and that allows the arbovirus to complete its life cycle. What happens after that point is probably not particularly important to the arbovirus. Well, what happens in the vertebrate is the vertebrate brings the blood virus under control. And in the case of this mouse with this particular seminicuporous virus, the virus then gets into the brain within 24 hours, and the virus replicates in the brain. And then the immune response of the mouse brings the virus infection in the brain under control. So that's a typical model system. We inoculate the mouse. These mice actually don't look particularly ill with this strain of the virus, but they do have a lot of virus in their blood, as you can see, and the virus replicates in the brain. So the first line of defense is the interferon system. This is a molecule that is produced by virus-infected cells. And if we have mice that have no interferon system, so green now is mice without an interferon system, you can see that the virus titers in the blood go even higher, 100-fold higher. So now we're at 10 billion infectious units per ml of blood, and the mouse dies. So interferon is something that protects most of us all of the time from virus infections. It's the first line of defense. And if you don't have it, you're going to get pretty damn sick. And people with genetic diseases that affect their interferon system have to live effectively in a bubble so they don't get any infections. So what happens when the virus gets to the brain in our mouse? So this is a section of a mouse brain. And what we can see here in this section of the mouse brain is all the dark areas are where the virus is, and the virus has come from the blood into the brain. And here's a histopathological view of that. So here's a blood vessel, and that would be one of these dark spots. In the middle of one of these dark spots is a blood vessel. And the virus has come from the blood into a cell. And you can imagine these black dots here are virus particles. They're not, but they are in effect a virus-infected cell. So this virus-infected cell here is giving out lots of virus potentially, and that will infect other cells. And then what happens is lots of these dark nucleated cells around the blood vessel here. These are lymphocytes. These are your immune system cells. These come out of the blood, and they come into the brain, and they cause an inflammation. And it's the combination of the virus infection and the inflammation which make people or animals sick. And this is known as encephalitis. Let me go to talk for a moment about basic virology. So a virus is not like a bacterium. A bacterium can replicate out in liquid. So it could replicate in your milk that's going off in the fridge. It could replicate in a bit of stagnant water. Bacteria can do that, but viruses can't. Viruses have to get inside the cell in order to replicate. So they are dependent upon the structures that the cell has. So here's our virus. It gets inside the cell, and then it replicates, and the virus comes out and infects other cells. There's a huge story in there, but I'm not going into that today. So now these other cells are infected, and the infected cells make a series of molecules called cytokines and chemokines. And these are important because they change the state of surrounding cells and they call in inflammatory cells. So they spread out from around the cells that made them. And these red ones are interferon molecules. I talked about interferon. And when the interferon gets into the cell, or the interferon actually has a receptor which it binds to on the cell surface, once the signal from the interferon is transmitted to the cell, the cell produces an antiviral response. It upregulates hundreds of genes and makes an antiviral response. And these cells now become refractory to virus infection. What happens in most cells is that when they get virus infection, they actually detect that they're virus infected and they kill themselves. And that's an important point. They kill themselves because if a cell can die quickly, it won't produce virus. So when the virus gets into these cells, the cells detect it and they switch on a suicide process and basically they die. And if they can die quickly enough, there'll be no new virus. And then new cells will generate in the tissues. That's what might happen in your skin. That's what might happen in many other tissues. And it's what happens in the developing brain. So here are some sections from the developing brain, and they're stained for a marker for those that are up on these things, which is called caspase-3, which is a marker of apoptosis. And it shows that these neurons are undergoing apoptotic cell death in the developing mouse brain. And the developing mouse brain is very prone to, or some of the cells within the developing mouse brain are very prone to undergoing apoptotic cell death when they get virus infected. However, if we go to the mature mouse brain, so now this is a mouse that's over 3 weeks old, I'm calling a mature mouse brain. It probably requires to get to 8 weeks before it's fully mature, but it's mature from the point of view of what I want to say today by 3 weeks or 4 weeks. And here we've got some mice that have severe combined immunodeficiency. And now these mice, without any immune system, they can't control the viremia and they can't control the virus in the brain. And these foci of infection in the brain, I've used the same picture, but these foci of infection in the brain now will remain essentially for the life of the mouse. And these adult neurons don't die. They can't die. They've evolved not to die because adult neurons are for life. Your adult neurons, my adult neurons, will hopefully live for 80 or 90 years, the same cell, without dividing, which is unlike all the other cells of our tissues. So these cells don't undergo cell death when they're virus infected. And the virus remains in the cells without any inflammatory response in some cases and just sits there. Here's a brain from a mouse. You would think it was just a normal mouse. The mouse looks normal, behaves normal. It was infected with this virus at birth, and it grew up, and the mouse was totally normal. You take the brain out, you show it to a pathologist. They say there's nothing wrong with this mouse brain. Everything's normal. But you do a test for the virus, and huge numbers of neurons are infected with the virus in the brains of these mice. So the mature neurons don't necessarily die, which is an important point, and led us many years ago when I was in Cambridge to talk about altruistic cell suicide and the specialized case of the virus-infected nervous system. And that is that cells will die when they get virus infected, but neurons don't. And that explains why a lot of viruses can persist in neurons. So I'm going to talk now about a couple of different projects that are happening in the lab that we're looking at this in a bit more detail. So one of them is around interferon, and Divya Narayanan in the lab has been looking at interferon in mouse brain cultures. I'm not going to go through the data here, but I just want you to see the kind of things that Divi has done. And she's been looking at the titers of virus in the brains of mice, and she's been looking at what effect interferon has on that. And then she's looked at all of the 30,000 genes that the mouse has and looked at all of the expression of those genes and how they're altered when you infect the mouse, or the mouse neurons in this case, or you give interferon to the cells before you infect them. And what Divya found was that immature neurons don't produce interferon, but they respond to it. And there's a differential expression of genes between the types of neurons, and that the immature neurons do not transcribe the genes that are able to switch on the interferon system and many other defense systems. So immature neurons die, but they also can't switch on the defense mechanisms. So that's one reason why these arboviruses and other viruses are particularly pathogenic in the developing nervous system. Damien Pfan's another PhD student in the lab, and he's looking at just one of the hundreds of interferon-induced genes, and this is RNase L. And in this case, there's an interesting story in that RNA molecules in cells can either be degraded by RNase L if they bind a molecule called TTP, or if they bind a molecule called HUR, they're protected from this RNase degradation. So guess what happens when the virus infects cells, and here are some cells that are not virus infected, and red is this molecule called HUR. You can see this molecule HUR is now distributed right throughout the cell cytoplasm and the nucleus. But within 8 hours after infection, it ends up in around the outside of the cell in the cytoplasm and not in the nucleus. And this is because the virus now binds the HUR molecule in order to stabilize its transcripts and stop them being degraded by the RNase L, which is an interferon-induced gene. I'm going to skip through that and come to Lucas's work. And we found some time ago that the Semliki-Forest virus can persist for long periods in the brains of mice, and this includes immunocompetent mice. And I made the point about the fact that the neurons can't die. But why is the virus persisting there? Well, possibly just because the neurons can't die, but maybe also the immune responses in the brain are switching off too soon. So this is where we got to talking about immune responses. Here were the infected cells, and these infected cells were making cytokines and chemokines. And those chemokines call in other cells. These are immune cells now that come in. I showed you them around the blood vessel. And these immune cells, some of them make antibodies, and the antibodies get rid of the virus. And they make additional cytokines, and they might call in other cells, these yellow cells. Some of these cells are killer cells, and they will kill the virus-infected cells. And I should add that Peter Doherty won the Nobel Prize for discovering how that particular bit happened. And these cells will then die and fade away, and the tissue will recover when these molecules called suppressors of cytokine synthesis come in and switch off the cytokine production. So we wanted to know what was happening with these suppressors of cytokine synthesis in the brain. And so we looked at 2 of them. These are called SOX, suppressors of cytokine synthesis. Here are the cytokines that are being made and the chemokines. And Lucas has looked at these and can look at the amounts of each of them. And you'll see that here, if we just look at day 6, this is a wild type mouse. And this is a mouse that has got no SOX4 molecules. And you can see that it has got many more cytokines. And if we look at the cells that come in, there are all kinds of different cells that come into the inflammatory response. But again, if we look at the wild type mouse and then we look at the one that doesn't have any SOX4, then this has got many more cells. So these cytokines are important for suppressing the inflammation. So if we just look at the SOX4 deficient mice, There's earlier clearance of infectious virus from the brain if the SOX molecules aren't there. In other words, the immune response is more vigorous. The brain cytokine levels are increased, and there's increased inflammation. So this SOX molecule is important in dampening down inflammatory responses in the brain. Now, I showed you that because I wanted to give you a flavor of the kinds of things we're looking at. And that includes looking at the interferon response, looking at the inflammation, looking at how inflammation is dampened down in the brain, because that's important in encephalitis, because often people and animals are getting sick because of the extent of the encephalitis, the extent of the inflammatory response. So understanding the inflammatory response is important. I want to hand over to Julio now, and he's going to tell you in the last 10 minutes about mosquitoes and virus infections in mosquitoes of these arboviruses. We're going to have to swap the sharing though. So Julio, over to you. But let me introduce Julio first of all. He's a postdoc in the lab, and Julio did his PhD at the University of Edinburgh with Alain Cole, who was a Wellcome Trust Fellow in my group in Edinburgh. And then Julio went with Alain to the University of Glasgow, and then came to Australia to work at the CSIRO's Australian Centre for Disease Preparation down in Geelong, and then joined me at the Peter Doherty Institute. So, Julio.
Yeah, thanks for the introduction, John. So my talk is a bit different from John's. I'll just be talking about what's going on in the mosquito, and how mosquitoes respond to viral infections, and how also we can use those viral antiviral mechanisms to control the spread of viral diseases. So as John said, we're based at the Peter Doherty Institute, and we are part of the Faculty of Agriculture and Veterinary Sciences. So mosquitoes are the deadliest animal in the world. Someone calculated that around 1 million humans a year get killed by mosquitoes. I don't know how they did this calculation. Ironically, the second deadliest animal to humans are other humans, which kill 500,000 people a year. And the third prize goes to snakes, which kill 50,000 people a year. So mosquitoes are important when controlling human and animal diseases. So in this presentation, I will just give you an overview of what happens when a virus infects a mosquito. I will also tell you about the mosquito immune responses against those viruses. I have a couple of slides about some projects that we are doing and that we finished, and then some take-home messages from this. So first, when a mosquito bites a virulent host, I don't want you to think about the mosquito as a syringe. It doesn't suck the virus and then spits it out. It doesn't work like that. It's a bit more complicated than that. So if we look at that section of the mosquito, we have the salivary gland here. We have the crop here. This is where they digest sugar, because mosquitoes drink nectar. They don't feed on blood. And here is the midgut. That's where the blood goes for female mosquitoes that they need in order to lay eggs. So what happens when a mosquito takes a virulent blood meal? So the virus and the blood go into the midgut. The blood gets digested there, and then the virus goes to the epithelial cells of the midgut and infects some foci of cells in the midgut. And then those foci of cells can produce viruses, more viruses that they spread, and they systemically spread to the mosquito. I have to mention as well that unlike in vertebrates, when mosquitoes just get infected, they don't really die. They don't suicide like we saw with vertebrates. They produce a lot of virus in the beginning, and then they just control the infection, and they just produce like a little basal amount of virus. And the virus will be in the cell until the cell dies. The cell can't get rid of the virus. Anyway, so as I said, these foci of cells produce some virus. The virus systemically spread, and once in a while there will be some virus that goes all the way to the salivary glands. It will infect the salivary glands tissue, and salivary glands have a special environment which allows them to produce a lot more virus than normal cells. So that virus will be stored in the salivary glands, and then when the mosquito takes the next meal, that will be split there, and that's how you infect the next host. So obviously, the virus is still a pathogen for the mosquito, and the mosquito is trying to get rid of the virus. And how does the mosquito immune system work? So, I mean, they've been writing books about this. So basically, there are several pathways. There are human selling pathways, like in invertebrates, that control viral infection. I'm not going to go into them because I have 10 minutes to give this talk. So the bottom line is that the most important pathway is called RNA interference. So this pathway, how it works is that a virus infects the cell, it releases the RNA viral genome. This genome gets transcribed by a virus RNA-dependent RNA polymerase and produces this double-stranded RNA. So double-stranded RNA is foreign to eukaryotic cells. Eukaryotic cells don't have this. So it becomes a kind of alarm, an epitope of alarm. So there's this molecule called dicer, dicer2, that can sense this and sense this is foreign. So what dicer does, it cuts off this chunk of double-stranded RNA and integrates it. This chunk is 21 nucleotides. It's always 21 nucleotides. Then dicer makes like a bit of a hairpin with this molecule of RNA and then integrates it into this complex called the RISC complex. And then the RISC complex goes around the cell and it keeps probing RNAs around the cell, random RNAs. So then when the RISC complex finds an RNA that exactly matches those 21 nucleotides, it clicks it. And because this RNA is derived from the virus genome, that's how the cell gets rid of these viral genomes. Right, so that's like the basic major system. What happens if we remove this? Or we can remove this by using transgenic mosquitoes or by silencing this pathway. So the mosquito dies because of the viral infection. So this pathway seems to be the most important. There is a derivation of this pathway, which is becoming later important for this talk, and it's kind of become a bit flavorsome in the past couple of years, which is that it's called the pre-read pathway. So this is a bit different. So again, the virus produces an RNA genome. That RNA genome gets reverse transcribed by endogenous reverse transcriptases on the mosquito. So the RNA becomes DNA, and then the DNA becomes integrated to the genome of the mosquito, which is very interesting. So then that DNA can produce more RNA. That RNA can make double-stranded RNA as a template. And as we see before, double-stranded RNA can destroy this viral double-stranded RNA. So now this is very interesting because if you have a mosquito and that mosquito gets infected by different viruses, all these viruses are going to end up in the DNA of the mosquito. So if you have a mosquito line and you sequence the DNA, you can see that all the viruses that were infected in the line in the past. It's quite interesting. Now, I'm gonna tell you a bit about laboratory projects that we're doing. It's very fun to work with mosquitoes, and we do most of our experiments are done in Bio21 because they have an insectory and one insectory. So this is a project that my student Matilda did. So what Matilda was looking at is something called superinfection exclusion, which means that if you have a mosquito, if you have a mosquito and you infect that mosquito with a virus, alpha virus, like Semiti or Ross River, and then once the infection is passed, then you try to reinfect the mosquito with a related alpha virus, then Matilda was looking at what's going to happen. Either the first alpha virus blocks the second, they both mix up, or the second one displaces the first one. As well, what he found was very interesting. He found that basically you get a reduction of the second alpha virus that comes in, a very high reduction. So this is important because let's say, as a future hypothetical strategy, you could infect a population of mosquitoes with something that is not pathogenic. And then when a pathogenic virus comes along, then it can infect a population of mosquitoes. So that's one of the projects we've been doing. The other project we're doing has to do with vertical transmission of antiviral immunity. So what I've observed was that if you give a virus to a mosquito and the mosquito becomes infected, that's why it's all blue now, then you rear that mosquito. So the mosquito lays eggs, makes larvae, and the larvae grow and then you challenge that offspring with a virus, that offspring is protected against that virus, right? So here we have, this is the graph, the same graph that John was showing. So this is your titer, your amount of virus in log scale. So for example, at day 5 or at day 7, you have basically 100 to, well, 10 to 100 to 1,000 times less virus in mosquitoes whose parents were infected with viruses. So then I started to look more into this vertical transmission of immunity, and I tested how many generations this lasts for. So if I infect a first generation of mosquitoes with the virus, then I test if the offspring can become infected, and then I can test if the offspring of the offspring can become infected, and then again and again. Every time this happens, I'm not infecting the mosquito, I'm just giving the blood without any virus. And what I could see is that the immunity against this virus can last several generations. So if you infect, the grandparent mosquitoes, the grandchildren of those mosquitoes are slightly resistant to these viruses. So for example, as we saw, this is the first generation, you get like 10 times less virus. And the second generation, you get slightly like 5 times less virus. And then here you get 3 times less virus. And after the 4th, 5th, and 6th generations, there is no protection. So immunity has a significant impact reducing viral titers after 3 generations, which is quite important. So then I'll try to look at what's the mechanism of this and what triggers this. So I gave the parental mosquitoes either a viral replication particle, which is not a whole virus, it's a bit like the viral RNA vaccine, the mRNA vaccine. So it has a piece of RNA that replicates and expresses viral properties. It's a bit like a vaccine. Or I gave them virus which has been irradiated, so the RNA genome doesn't work. So what I saw is with the RNA replication particle, the viral replication particle, I get protection. However, when the virus RNA is damaged, we don't get any protection. So this system requires actively replicating virus, which is quite interesting. So then going back to the second pathway that I was showing you, this DNA integrated pathways, one of the key components in this pathway is this reverse transcription enzyme. So well, maybe we should look into that. And another component of this pathway that I haven't explained is that once the genome is integrated, this viral RNA can come out from the genome. It can go into an exosome, and it can be sent to another cell, and it can be integrated in another cell. And this cell can also produce this viral mRNAs, can protect this cell. So now this cell here is protected. So this can travel through all the body of the mosquito. So I thought, well, if it can travel through the body of the mosquito, could this be sent to the next generation? So I said, well, what happens if we basically shut down this protein, so then this doesn't happen? So because obviously viruses like HIV have all these retroviral properties, I managed to get a drug called ACT. This drug is an anti-HIV drug which shuts down this reverse transcription state. So what I did is I fed this to mosquitoes, and then I infected them with virus. And as expected, there is something like 10-fold more virus in mosquitoes who took the drug. So that system that I showed you before seems to be very antiviral. Now, if I look at the offspring of these mosquitoes, the whole vertical transmission immunity is reversed. And now these offspring don't have the protection because of the parent mosquito lacking that enzyme or having the enzyme inhibited. Even more, if we look at generally alpha viruses don't get vertically transmitted. If we look at the offspring of that mosquito, you can see that the parents can send virus to the offspring, which was also very interesting. So now just the conclusion, take home message. So mosquito antiviral immunity, the mosquito immune system, it was thought to be an innate immune system. I think we're starting to believe there is more adaptive immune system, and it can be inherited, which is kind of adaptive immunity. Different strategies like superinfection exclusion can shut down incoming alpha viruses if your mosquitoes are infected already with a virus and is related to them. Vertical transmission of immunity. So mosquito derived from infected parents are more resistant to the same or related infection in the offspring. And then vertical transmission of antiviral immunity. So this can be shut down with antiretroviral drugs. So we think this whole mechanism is related to this reverse transcriptase system that the mosquito have. So why is this important? So ultimately, what we want to do is develop strategies to shut down, to block viral spread through mosquito populations. So you can imagine this is a very hypothetical case. If we get a population of mosquitoes and we vaccinate them with these viral replication particles, this whole population and then the offspring from those populations will be refractory to virus. And then in theory, you could, these naturally occurring mosquitoes won't be able to transmit, let's say, Ross River to the human population in that area. So that's what we were kind of hoping. So in the future, hopefully we can reduce the number of the 1 million deaths of people caused by mosquitoes using these antiviral strategies. And I think that's the end of my talk. And I pass back to John.
Thanks, Julio.
No worries.
So let me just share my screen for a second, and then we'll finish. And can you see that? Hopefully, yep. Okay, so I want to summarize what we've just said. So in summary, arboviruses are responsible for many animal and human diseases. Some are endemic and some are epidemic. Encephalitis, haemorrhagic fever, and arthralgia are important, and there are vaccines for some arboviruses. Generally, though, there are no cures for virus encephalitis, and we need to better understand the pathogenesis of brain inflammation, which we're working on. And these and other viruses may persist in tissues. And mosquitoes transmit many of the medically important arboviruses. We've also worked on ticks. We've not talked about ticks today. And we've discovered that mosquitoes have inheritable specific immunity, and Julio's just talked about that. So I'd like to finish by just acknowledging the people in the lab, Julio, Lucas, Divya, Mathilde, Damien, and Isabel, and our collaborators, Catherine Kedziersko, Jason McKenzie, and Ori Hoffman, and Prasad Paradkar at the CSIRO Australian Centre for Disease Preparedness. So thanks very much. Sorry, we're a little late, I think, but I'll stop sharing and we'll take any questions.
Thanks very much, John and Julio, for a very, very nice presentation on a whole range of important aspects relating to virus infections. There are a number of questions. Nicholas Deutsch has a question in relation to vertical transmission. Nicholas, can you ask that? Or did you want me to read out your question?
Yeah, sure, Robin. Julio, I was just interested if this vertical transmission of immunity has been documented in other animal species like you have.
Oh, what do you mean other animal species? I think it was kind of hinted in Drosophila, but it wasn't with Drosophila, with some of the Drosophila viruses, which are quite deleterious. So Drosophila tends to die from these viruses. But to our knowledge, it hasn't been describing mosquitoes, especially as antiviral. Yeah, if that answers your question.
Yeah, that's great, Julio. And higher order animals, has that kind of vertical transmission of immunity been seen at all?
So the RNAi system, I mean, there's a bit of controversy. Some people say that the RNAi system also works in humans with the shRNA. I'm not sure. I'm not an expert there. It's pretty controversial. We know that in humans, you have provirus in your genome. So ancestral viruses that have been integrated there. Not sure if they play such an active role as they do in insects, though. I would say maybe not.
I think that's right. It hasn't really, there's no evidence of that as far as I'm aware in mammalian systems. Although there were some interesting papers some years ago by Rob Sinkernagel, a Nobel laureate, about the fact that some infectious disease genomes may get reverse transcribed into DNA in mammals, and that might be important in immunity. So I'm not sure what happened to that work, but it might be worth looking in there if you're interested in that topic.
Okay, Neil Young has a question in relation to variation in immune responses. Neil?
Hey, thanks for the great talk. Just looking at some of the projects in the faculty over the years, we know that non-model organisms are terrible to work on because of their variation in their immune systems. And just seeing some of the plots that Julio had on the mosquitoes, I guess there's some variation in immune response to mosquitoes. And one thing that we do in our projects is try and inbreed as much as possible to keep things simple. And I know that in the mouse world, that's the case. Do you keep that in mind in your insectorium? Do you go for inbreeding and simplicity, or do you try and capture what would be a natural variation when talking about immunity?
So we try to do the natural variation. So most of the mosquitoes are within 3 or 4 generations of wild-caught mosquitoes. So they are not domesticated quite that well. So those lines are pretty wide. They're called in Queensland, these are Aedes aegypti mosquitoes. And even in laboratory lines, you still get a very big variation. That's why you use maybe at least 50, if not more, mosquitoes per experiment. Some mosquitoes might not get infected at all. Some mosquitoes might have very low infection, and some of them might have very high. So I think it comes to there are several factors, but it comes to an individual mosquito state. So it's good to have high numbers so you can kind of get a picture of what's going on. Hope that answers your question.
Yeah, great. Thank you. Thank you.
Glenn Browning also has a question regarding vertical transmission. Glenn?
Yep, thanks, Julio. That was a great talk, and John. I was wondering whether that vertically transmitted immunity that you're seeing in mosquitoes can be, I guess, boosted in inverted commas by re-exposure to the virus while the mosquito is still partially resistant.
Yes, so, well, I didn't go into it. So some of the graphs I was showing, especially when I was using BRPs, so those are mosquitoes that lost their immunity after the whatever 5th, 6th generation, and then I kind of gave it to them back again. It gets boosted, yes.
And does, if it's boosted, can you achieve a higher level of resistance than? So there's a limit,
the same
level of resistance. Yeah, okay.
Yeah, just the same. I think that doesn't go. Maybe there's a threshold, so it doesn't go above.
Okay, thank you.
Thanks so much. I was wondering, John or Julio, how good is the organoid system to how genuine is it to look at responses and study virus cell interactions or host interactions? You know, I'm thinking in particular of brain organoids that are currently being used as model systems.
Yeah. I mean, it's an important point, Robin, and the cell system that one chooses is often really important in these kinds of studies, and in no system more so than the brain. Because actually, if you want to study what happens in mature neurons, it's pretty damn difficult to do in culture because mature neurons, by definition, don't divide.
Yeah.
And so all cell culture work is pretty much based on the fact that you've got a cell line that will divide. So the neuronal systems rely on a couple of things. One, cell lines that can be differentiated with particular growth factors in culture. So for example, some neurons, neuronal cell lines, you can give them nerve growth factor and they convert from being a replicating cell to being a static cell. Or you can do what we did, which is to take the neurons out of the developing brain, in our case a mouse, and culture them. And if you culture them for a week or so, they differentiate in culture of their own accord if they're primary neurons. And so we compare them at just after they're put into culture, and then a couple of weeks later, and those are the mature neurons. And the same is true of the mosquito cell systems. There are mosquito cell systems, but they tend to be derived from mosquito larvae. And so yeah, choice of type of cell that one works on is very important, definitely.
Well, thank you both very much. That was really, really interesting. Certainly a lot more work to do, obviously.
Yeah, there's huge amounts to be done on arboviruses, and now Japanese encephalitis virus is here in Australia. Perhaps it will generate a lot more interest.
Thanks so much, John. Thanks, Julio. And thanks to all of you for attending today's seminar session. Just to let you know that there's the next seminar is on the 12th of April from memory, and Prem Bala will be presenting on developing crops for future climates. So we hope to see you then. So thanks very much again to everyone for attending today's session.